Okay, so this is going to be the last chunk of material of a break that you're going to be looking at. There'll be two PowerPoints, which is only half of the gene expression stuff that we will talk about. So when we get back, we will resume talking about this content. So Monday you were looking at meiosis. Wednesday you were looking at protein synthesis, and then today we are starting to talk about gene expression, which will segue into genetics. And from there, we're really just wrapping things up. It's going to move very quickly before the actual test. So the first thing we're going to talk about is the prokaryotic regulation of genes. And remember that genes are pieces of DNA that control a certain trait. Basically, for example, for each gene, there is certain regulation. And so each gene creates a specific protein. So in this case, tryptophan is a protein, specifically a hormone that is made, and there is regulation of how much of that I'm making. The two ways that can happen is I produce something that will interfere with the function of the enzyme in that pathway. So an enzyme is required to read that gene and make the protein, so I make something that gets in the way of it being read, or I can produce a gene regularly that can inhibit the transcription of one of those enzymes, so I don't even make the enzyme to read it. So in one type of regulation, I will just stop the enzyme from working. In the other case, I stop that enzyme from even being made, so I can't make that protein. When we talk about eukaryotic cells have way more genes, basically 23,000 in human cells, and on average, prokaryotic cells only have about 3,000. Physically, there are more obstacles to regulate eukaryotic genes because there's so much more DNA to manage. And so it's a far more complicated system. In addition, there are other non-histone proteins that are used in eukaryotic gene expression that are not used in prokaryotes. So essentially what this is telling us is that the reason we start talking about this with prokaryotes first is that it is a simpler process because there's less DNA to deal with. So the term operon is going to be really important. Operon is a group of prokaryotic genes with a related function, and they're often grouped together and transcribed together. So in addition, the operon only has one promoter region for the whole operon. If we remember, the promoter region is where we start reading it to make RNA to make the protein, right? So my DNA has a promoter where we start reading it. And so operons are going to be sequences of genes that create proteins that all have a probably joint purpose. And so we can see that here. An operon is usually composed of the following things. Structural genes, so genes that are related and used in a biochemical pathway. So they have a series of genes of the operon that code for certain proteins that are used for something. They will always have a promoter, which is the nucleotide sequence that is at the beginning of that gene that binds with RNA polymerase to start the creation of RNA, which will end up making the proteins. Then we always have the operator, which is the nucleotide sequence that can bind with the repressor protein to inhibit transcription. So if I want to stop making this series of proteins, I have my operator, which is where it can be bound to, that will stop the production of these proteins. We also see things called regulator genes, and this gene can produce a protein called a repressor that inhibits the transcription of an operon by attaching to the site. And so it usually will attach to the operator site, and it represses the creation of that. So when we're talking about whether or not this gene is going to be made, we have to think about a couple other things. Modulators can bind to the repressor at a site that will basically activate or deactivate the repressor. And then the repressor will in turn stop the transcription of something from happening. When it talks about allosteric properties, allosteric means it can either start things up or it can stop things. So in this case, a modulator would be, could bind to the repressor and stop the activity. It could leave and make the DNA be turned into RNA. So an example of an operon that you'll hear about a lot is the lac operon, and the idea of lactose, or the digestion of lactose. If we look at the DNA strand, we see that the operon is a series of multiple genes, specifically your LAC-Z, LAC-Y, and LAC-A genes. They're going to create 3 specific proteins that all work together to work with lactose. We'll also see that we have our, if we go back, right, we have our promoter and our operator. We see that our promoter, right now, the RNA polymerase is around that, and our operator would determine whether or not the it's going to be read or not. And so that's what it looks like, the lac operon. Is this animation going to work? Oh, here we go. Is it? Yeah. So basically what they are showing is that we have a regulation gene before the actual operon. The regulation gene will either produce something that will make the repressor active and it will stop it, or it will make something that will bind and actually make the DNA get read. In this image that's happening, we see that the genes are actually being read, or the codon is being read, and it makes a series of 3 proteins that work together. So again, it's going to go through it one more time. The regulator gene makes mRNA. It makes the active repressor protein. If allolactase binds with that repressor protein, we'll see that it's not going to be able to move and stop the reading, right? So that means that if lactose is present, it's not going to get in the way. If lactose is not present, it's going to bind to the operator and stop the transcription. In this case, lactose was present, it bound to the repressor, and so the repressor couldn't bind to the codon, and I make my protein. The absence of lactose in the lac operon. If there's no lactose or allolactose, like we talked about, we see that the regulatory gene makes the mRNA and makes my repressing protein. Because of that little half circle, lactose did not bind in there, so it doesn't change the conformation or the shape, and it goes and binds to the operator. And so RNA polymerase, even though if it binds to the promoter, it can't read everything, therefore prohibiting transcription and translation. This is an animation of the opposite. I'm going to assume that we don't need to see this necessarily, but without the presence of lactose or allolactose, we're going to see that it's going to bind, and that RNA polymerase doesn't end up binding to it, or it can bind, but it can't physically read it, and that's the end of that. The synthesis of tryptophan and the repressible TRP operon. Again, if we look at the strand, we see we have our TRP repressor. That's going to make the repressing protein if it, so we can stop the process if we need to. We see that we have our promoter, where RNA polymerase is going to bind. We have our operator, which is where the repressor will bind if it can. We have our sequence of genes, which is the actual operon, and it works basically the same way, right? So we know that if TRP operon with no tryptophan present, in this case, it's actually going to do the opposite of the first one. So the first one, when lactose was present, it didn't inhibit it. In this case, if tryptophan is not present, it won't inhibit it. And so it really depends on gene to gene which way it works, but just the opposite is true. the thing that we need to remember is that there's always a regulatory gene, which makes a repressor protein. There's always the promoter, which will start transcription. There's always an operator, which, if bound to the repressor protein, will stop transcription. And then we have our operon, or our series of genes that make the protein. Again, this one shows that if tryptophan is not present, it can't bind. If it is present, then it does bind. Therefore, we can assume that an enzyme like this, if I have too much of it, it's going to stop making it, right? Whereas if I don't have any, it will start making it. The LAC operon and the TYP operons are examples of negative gene regulation, as the repressor protein inhibits transcription. And so in both cases, the repressor protein, once it binds, it keeps the transcription from happening. In the LAC operon, I need lactose for it to bind, and the the opposite is true of TYP. The example of positive gene regulation is that the opposite is true. Basically, it requires something else to bind to it to make the process happen, whereas the past two, when something bound to it, it halted the process. For this specific process, which is again the LAC operon, when lactose is present, the repressor doesn't bind, so transcription can happen. The other thing that actually has to happen is at the CRP binding site, there's going to be a certain enzyme that binds there that will make it transcribe. And that's going to be the end of eukaryotic gene regulation. What you need to know about this is that if you understand the simple structure of the operon and the steps, you should be okay. Please ask questions about this when we get back, if you need to.